If you work with mouse mesenchymal stem cells, or MSCs, you may know how difficult it can be to isolate and culture these cells. Mouse MSCs occur at very low frequency in adult mouse bone marrow, and, to further complicate matters, hematopoietic cells often contaminate cultures and may persist in culture even after several passages. Studies have shown that in the adult mouse, the CFUF frequency in compact bone is significantly higher than bone marrow. This suggests that the major reservoir of CFUF is the surrounding bone tissue, rather than the marrow itself. Stem Cell Technologies has developed this instructional video to help researchers achieve cell preparations with high yield and viability of MSCs isolated from mouse compact bone. After the femur and tibia have been harvested, thoroughly clean the bones. This will help to minimize cell contamination from other sources. It is particularly difficult to remove the attached muscle and tendons from the epiphyses, and therefore we recommend discarding this portion of the bone. To do so, use a scalpel to remove the epiphyses. The bones can be efficiently cleaned by holding one end of the bone with forceps and scraping along the length of the bone with a scalpel. Place the clean bones in a mortar containing cold PBS containing 2% FBS and 1 millimolar EDTA. Crush the bones with a pestle using only enough force to crack open the bones. It is important to use the least amount of force possible. The goal is to crack the bones to release the marrow rather than to pulverize the bones. A loss of cell viability and excess debris will be generated when bones are harshly ground. Instead, apply downwards pressure to the pestle to fragment the bones. Using the pestle, gently agitate the bones to free the bone marrow. Remove the buffer using a pipette. The buffer containing bone marrow can be filtered through a 70 micrometer cell strainer and used for subsequent experiments or cell culture. Add fresh buffer and repeat the agitation and removal of bone marrow until most of the bone marrow has been removed. At this stage, the bone fragments will turn light pink to white in color, and the buffer will be colorless. Generally, this takes about 3 to 6 washes. Transfer the bone fragments to a 100 millimeter dish. Add 2 ml of 0.25% collagenase type 1 in PBS containing 20% FBS. Ensure the bones are completely covered in solution. Let sit for 3 to 5 minutes at room temperature. As collagenase activity varies widely, Stem Cell Technologies provides pre-screened collagenase to ensure consistent performance. Using a scalpel, chop the remaining bone fragments into fine pieces 1 to 2 millimeters in size. Proper bone fragmentation is required so that the subsequent enzymatic digestion releases the maximum number of cells. Digest the bone tissue with collagenase for an additional 45 minutes at 37 degrees Celsius in a shaking water bath or a clean bacterial culture shaker. Filter the cell suspension through a 70 micrometer cell strainer. To maximize cell recovery, wash the bone fragments with buffer and again filter through a 70 micrometer cell strainer. To collect the cells, centrifuge at 300 G for 10 minutes. Resuspend the cell pellet in an appropriate medium. Your cells are now ready for use. At this stage, small particles and debris may be visible in the cell suspension. You can expect to obtain 1.5 to 3.5 times 10 to the 6 cells per mouse. If the cell yield is greater than 5 times 10 to the 6 cells per mouse, this is an indication that the marrow was not sufficiently depleted during the flushing process. To achieve efficient expansion of your mouse MSCs, culture your cells in mesenchymal proliferation medium under hypoxic conditions. If you would like to enrich mesenchymal stem cells from your cell population, Stem Cell Technologies' EasySet Mouse MPC Enrichment Kit can be used to deplete cells of hematopoietic origin. To learn more about Stem Cell's range of products to enrich, expand, and differentiate mouse mesenchymal stem cells, please visit stemcell.com.